Hello and welcome to the Florida Friendly Landscape Program's Homeowner Webinar Series. My name is Jen Marvin and I'm the Florida Yards and Neighborhood Statewide Coordinator. Today we have Blake Costine speaking with us about medicinal plants. Your microphones have been muted. If you have questions, please type them in the chat box and we'll enter them at the end of the presentation. I will answer them at the end of the presentation. Please stick around until the end to take the exit survey. It helps me give you the kind of programming you like and lets us know how we're doing. Our next presentation will be June 15th at 11 a.m. Eastern on attracting wildlife to your yard. I'd like to introduce today's speaker. Blake Costine is a master's student in the Environmental Horticulture Department at the University of Florida. His research focuses on exploring Florida natives for plant-derived pharmaceutical compounds. He is a 2020 graduate in plant sciences with an emphasis on plant genetics. He was associated with both the College of Pharmacy and the College of Agricultural and Life Sciences during his education and is fascinated to combine both these two interests for his graduate research. His long-term goals are to pursue a horticultural research career in academia. Before this webinar, I would like to note that the information given in this presentation is for educational purposes only and should not be construed as medical advice. This is very important. So without further ado, I'd like to turn it over to Blake.
Hello, everyone. Let me get my screen shared. Okay, good morning, everyone. Background about medicinal plants, and also talk a little bit about my research and some other groups here's research here at the University of Florida. And then lastly, cover a few plants with some medicinal properties that can be grown here in our Florida landscapes. So I'd like to reiterate that the information presented here today is not a guide to using plants for medicinal or other purposes. If you have any medical issues or require treatment, please seek the advice of a licensed medical professional. The information in plants I described today are only for educational purposes and maybe to give you some idea of some medicinal plants you could add to your landscape. So why should we grow plants? Well, because they make us happy. Studies have shown that people at work who can see plants report significantly greater job satisfaction than those who can't. I feel like this is a good excuse to use when your boss questions why your office looks like a small jungle. So our home is abundant with diverse plant life that is the key to supporting all of the beautiful life forms that live amongst us. As of 2016, we've only explored 15% of the chemical diversity of plant life with over 350,000 plant species. We have only begun to scratch the surface. Plant metabolites is where we see this chemical diversity being derived from. Primary metabolites being essential for plant survival and is most common among the plant kingdom, but the specialized metabolites is where we begin to see this large chemical diversity. They address the special needs of plants and are often specific to a specific genus or species and are localized in certain tissues. So a little more about primary and secondary or specialized metabolites. These primary metabolites produce structures such as sugars, amino acids, proteins, nucleic acids, and polysaccharides, and these are responsible for carrying out most of the needs that the plant needs for life. They drive cell division and growth, respiration, store nutrients, and help in reproduction. Although the specialized metabolites being where we see this large chemical diversity, plants produce metabolites such as phenolics, alkaloids, saponins, terpenes, lipids, and carbohydrates. So why do we see this chemical diversity? Plants create a diverse group of chemicals in order to interact with the organisms in the world around them. Plants create these chemical components in order to attract or repel insects and animals, to signal to other plants, or to defend against fungi and other plant pathogens. These chemical components can be nutritious, potentially poisonous, or medicinal for humans when ingested. Humans have learned how to harness this chemical diversity of plants for a variety of our modern needs. Starting at the most basic of being a large portion of our diet, the main active components of our medicine, the use of their fiber for our clothing, spices which flavor our foods, the active components of our insecticides used to deter pests from our crops, and dyes that color the world around us, and lastly, a source of aromatic smells for our perfumes and air fresheners. Humans use approximately 32,000 plant species for a variety of purposes, ranging from food to materials to poisons, although our most common use is for medicine. Over 30% of the plants that we use to treat the ailments are used to treat the ailments of today's society. The diversity of specialized compounds aid our modern society in a variety of ways. This figure here from the journal Science in 2016 shows just a handful of important metabolites and their corresponding plants that produce them. Some common known metabolites, include cellulose, which is produced by cotton plant, which provides us flour to make our clothing and textiles, corn and other plants that produce beta-carotene that's converted in our bodies into vitamin A. Some lesser known plants such as Digitalis purpurea produces digoxin, which is used for treatment of congestive heart failure. Chrysanthemums produce pyrethrins, which are toxic to insects, and we commonly use these for our pesticides. And Taxus brevifolia produces paclitaxel, which is commonly used in chemotherapy for cancer treatments. So plants produce hundreds of compounds we use as medicines or drugs. Some kind of common ones that you might be somewhat aware of is that willow bark is our source of aspirin. Foxglove, as I mentioned just a moment ago, is a source of digitalis, which is used for those cardiac problems. Pacific yew is a source of Taxol, which is another cancer treatment. I'm sure many of us are aware of coffee and tea as a source of caffeine, which is a stimulant. And then also peppermint as a source of menthol, which is used in creams for pain relievers and also a big component of many cough drops. So how do we find these medicinal plants? Our ancestors from almost all cultures have looked at plants to help in curing their ailments. Fortunately, many of these cultures have recorded and preserved texts of this medicinal knowledge and their preparation for the knowledge of future generations. One of our biggest resources as scientists is to mine this ancient knowledge passed down from our ancestors to rediscover these medicinal plants and incorporate this knowledge into our modern pharmacology. This is referred to as ethnobotany. This is the scientific study of the traditional knowledge and customs of people concerning plants and their medical, religious, and other uses. With modern technology, we can now identify and isolate the pharmaceutical compounds produced by these plants. These ancestral plants also provide us a starting point to broaden our search for other medicinal plants that have yet to be discovered. Some of our most ancient writings that we have come from the Sumerians. At the time, paper writing technology had yet to be invented, and they wrote into wet clay for their writings. A Sumerian clay slab shown here depicts the oldest known written evidence for the use of plants for the preparation of medicines. Dating back approximately 5,000 years old, this slab described 12 recipes for drug preparation and refers to over 250 plant species. Some medicinal plants, including poppy, henbane, and mandrake, were mentioned in this clay slab, and these are still used for their medicinal properties today. Also known as the father of medicine, Hippocrates expanded our understanding of medicine and medicinal plants, commonly credited with beginning the practice of medicine as a rational science and writing what is known today as the Hippocratic Oath. He provided knowledge of over 300 medicinal plant species and their subsequent preparations. Another important figure in history of our medicinal plant knowledge is Dioscorides. He was a Roman army physician for the armies of Roman Emperor Nero. And in approximately 60 AD, he wrote what is one of the most significant contributions of medicinal plant descriptions, De Materia Medica. Within 5 volumes, he described over 500 plant species and the preparation of over 1,000 drugs. De Materia Medica remained the standard text of medicine for the next 1,500 years. Today, we look back on our ancestors' knowledge in order to develop our modern medicines. Tu Yu's story is a prime example of this. She received the Nobel Prize in Physiology or Medicine in 2015 after using traditional Chinese writings in order to develop an effective treatment for malaria, which is still in use today. She extracted artemisinin from the sweet wormwood plant in the 1970s and found it was very effective in inhibiting the malarial parasite. She has a very noble and inspiring story where at one point she decided to volunteer herself for human clinical trials in order to expedite the safety evaluation process. This allowed for faster distribution of the medication to the people impacted by malaria. She was successful in her efforts, and her research was responsible for the survival and improved health of millions of people. Artemisinin is still being researched today to understand its mechanisms of action and to prevent future malarial strains that may become resistant to it. Here at the University of Florida, we have formed a joint team between IFAS and the College of Pharmacy. We call this group Medicinals and Controlled Environments, or MICE for short. Our team is focused on researching different plant-based products with medicinal properties. Some of our project leadership includes Dr. Paul Fisher from the Environmental Horticulture Department, Dr. Brantley Spakes-Richter from Plant Pathology, Dr. Roger Kellgren from the Mid-Florida Research Extension Center, and Dr. Satya Swathinatakaduti from Environmental Horticulture. So far, there are 2 main projects underway within our MICE group. In Dr. Swathinatakadudi's lab, I'm personally working with Scutellaria, commonly known as skullcaps, which I'll describe to you a little bit more about in just a moment. Dr. Paul Fisher and Dr. Selena Gomez's labs are also working on a project focused on ginger and turmeric. So a little bit more about the Scutellaria genus and my project. The Scutellaria genus is found within the Lamiaceae or mint family, and they're commonly referred to as skullcaps due to their dish-like calyx, which is found in their flowers, which is also where they derive their Latin name. There are approximately 350 species found within this genus, and they're distributed across Europe, United States, and East Asia. The majority of the Scutellaria genus are perennials, although some being labeled as annuals, and they retain common characteristics of other species within the mint family, including four-angled stems and opposite leaves. Most species within this genus retain similar flower structures, with 3 fused petals forming like the upper lip of the flower and 2 fused petals forming the lower. The flowers of the genus also range from blue to like a deep purple in color, and the leaf shapes of the genus vary widely, and they're often the only identifying characteristic for different species within this genus. This genus is also found in a range of ecosystems and habitats, ranging from semi-aquatic to very dry and arid conditions. There we go. In Florida, 13 species of Scutellaria have been identified, with 11 of them being native to the state. The 2 non-native species found in our landscape are Scutellaria racemosa, which is native to South America, and Scutellaria havanensis, which is native to Cuba. These Florida native species are found throughout a range of Florida ecosystems, ranging from sandy scrub to wetland areas. And as you can see from this map on the right, this genus is widely distributed across our state. With most of the members of this genus being found in almost every county, except for a few counties there in South Florida where Scutellaria has not been found. For the purposes of our research, we'll be focusing on these 3 species, Scutellaria erinicola, integrifolia, and lateriflora. The common name for erinicola is the Florida scrub skullcap. Integrifolia is commonly referred to as the helmet skullcap, and lateriflora is commonly referred to as blue or mad dog skullcap. So here are a few pictures of these native species. I've taken these are greenhouse grown plants that I've just taken pictures of recently, and you can see they all exhibit very different growth habits, leaf and flower structures. For the most part, Arenicola and Integrifolia have very similar flowers, but interesting enough, Lateriflora has these really tiny little flowers, and they're also found on the leaf margins of the plant. So the earliest known use for Scutellaria for medicinal purposes dates back to around 200 AD, specifically with the species Scutellaria bicalensis. So Scutellaria bicalensis is native to China, Russia, Korea, Japan, and Mongolia. And an herbal preparation that is still commonly used today in traditional Chinese medicine known as huangqin was described in Shennon's classic of Materia Medica. Huangqin is a root preparation of the Scutellaria bicalensis that is currently listed in the Chinese, European, and British pharmacopoeias. Huangqin is roughly translated to yellow golden herb, and I have some pictures here in a moment, and you'll see why it's translated to yellow golden herb. They have really like a deep, rich golden color to their roots, and is used commonly for fevers, liver, and lung treatments, and was even showing success with the author successfully using the preparation to treat a severe lung infection. Today, huangqin is still prescribed in many Chinese and Asian practices, aside from the traditional Chinese medicine. Scutellaria bicalensis has also been used in Chinese Campo medicines. Since this species has been extensively studied and utilized for its medicinal purposes, we'll be including it in our research and using it as a comparison to our Scutellaria native, Florida native species. So a little bit about some ethnobotanical use of one of our Scutellaria natives, Scutellaria lateriflora. So traditionally, Native American medicinal practices have used the aerial tissues of the Scutellaria lateriflora plant for different nerve disorders, including anxiety, epilepsy, and a few others, and is commonly referred to again as the blue or mad dog skullcap. And just something to kind of note here is that the Native Americans were using this particular species for its aerial tissues, while the Chinese traditional medicine cultures were using Scutellaria bicalensis for its root tissues for their medicinal practices. And that'll kind of come into play later on in some of my research. So with the Scutellaria genus, over 295 compounds have been isolated and identified, some of which have found a variety of bioactive and medicinal purposes. Particularly the flavonoids have become the main bioactive compounds of interest due to their high accumulation within the genus. Some of the flavonoids are also specific to the genus, meaning they have yet to be found in any other genus or any other genus within the mint family. And these unique compounds have been found to be the responsible component of the medicinal properties of this plant. So here are some of my research objectives I'm focusing on here at my time for my master's. The first is to explore the flavonoid metabolites in the 4 Scutellaria species using HPLC, which is high performance liquid chromatography. So I'm looking at the 3 Florida natives and the Scutellaria bicalensis as my baseline kind of comparison. And I'll get into a little bit in a moment of more about HPLC, and I'll describe that process to you in just a moment. Our second objective is to study the impact of light quality in controlled environments on the flavonoid profile. And then our last objective is to evaluate the growth and metabolite accumulation of the Scutellaria species in the field. So here is kind of a workflow process of my flavonoid extraction and analysis method. So first, starting off, I collect my tissues from my plants, either from greenhouse grown, or later on you'll see I'm doing a controlled environment study. And I collect these tissues from the plants directly and place them immediately into liquid nitrogen. So this is important because the liquid nitrogen flash freezes the plant tissues and prevents any changes in the metabolite profiles of these plants. So some of these metabolites are affected by plant stress, for instance, and maybe if I were to take some leaf samples and leave them on my bench for a little bit, the metabolites would actually change during that time. So after I've kind of collected my leaf samples, I will then place them in a mortar and pestle along with liquid nitrogen to keep them frozen, and I'll grind them to a fine powder. And then next I use methanol to combine with this fine powder and place it in a sonicator. So essentially the sonicator pushes these really ultra high frequency waves through this extraction sample, and it helps release the flavonoids from the ground powder into the methanol. And then next, I'll place this in a special auto sampler tube and place it into the high performance liquid chromatography machine. So a little background of how high performance liquid chromatography works is essentially we are able to take our sample and we push it through a very high pressures through a density packed column. And on the other side of the column, we have a detector. And at this detector, it's kind of reading the different compounds coming out at different points in time. So pretty much from that, I am able to develop a chromatogram kind of similar to this scene here. And this has these peaks where I'm now able to kind of see at what point in time these compounds are being pushed through the densely packed column. And I'm able to kind of now identify the flavonoids, and then now compare them to some known standards, and then be able to quantify actually how much there is. So this is how I'm kind of developing my flavonoid profiles for these different species. And so I'm able to kind of point out what flavonoids are in the plant, but also how much is in there as well. So to get our initial profiles of these different species, we grew them in the greenhouse setting and collected root, stem, leaf, and flower tissues. And I processed all of those separately to see if these compounds are being associated in different tissues. And kind of from our preliminary data, we've seen some relatively high concentrations of flavones in the Chinese skullcap's roots and in the leaf tissues of the blue skullcap. So again, this kind of pointing back to what I alluded to earlier, this matches well with the historical use of these medicinal plants, as the indigenous people that use these medicinal plants specifically use these tissues for their herbal preparations. So it's kind of interesting enough, we're finding the compounds that are attributing to being pharmaceutical to being in the tissues that the different traditional medicines are using for their preparations. Also, another kind of interesting thing to point out is I have purchased some Scutellaria products online, so from the Chinese skullcap and from the blue skullcap, and I was able to obtain some dried roots and some dried aerial tissues. And from this, we saw like really high increase in the flavonoid profiles, much more than I was seeing in my plants grown here. So I had kind of an initial hypothesis from this because the size and kind of diameter of the root tissues that I received in the mail were like really thick, almost comparable to this size here. But the samples that I was processing were really thin, and they were from very young plants. So these plants pictured here, these are the root tissues from Scutellaria bicalensis. These are much older, these are aged ones. So what I'm essentially doing is letting these plants grow out longer to kind of test this hypothesis to see maybe they're accumulating more of these compounds with age. So on to kind of my second objective. We're conducting a light quality experiment. So a kind of important thing to note here is the difference between light quality and light intensity. So essentially, light intensity is how much light the plants are actually receiving. Light quality is more of the wavelengths of light that the plants are receiving. So what I'm testing here is to see if a blue treatment will actually affect the different flavonoid profiles of these plants compared to kind of a more full spectrum white control treatment. So essentially, here's a picture of my growth room that I've set up. And I have my plants on my different shelves here, and I have my 3 blue treatments and then 3 control treatments. And I'm going to be studying, well, I have been studying Scutellaria bicalensis and Lateriflora, and I'm using 4 plants of each species to kind of test this out. I'm actually just now wrapping up this experiment and collecting my tissues now, so hopefully I'll have some interesting data to come from this. So we'll be interested to see if the flavonoid profiles are actually being altered or changed by this different light quality, since it's kind of known that different stresses are increasing the flavonoid profiles of these plants. And we're thinking that the blue light will likely stress these plants and increase how much of the pharmaceutical compounds they're actually producing. So on to my last objective. This is my field trial. So here's a picture of some of the plants that actually were just now put out into the field just recently. I'm studying those 4 species I was talking to you about earlier, the Chinese skullcap, and then the 3 Florida natives. And I'm evaluating them all for their general growth, their flowering, at what time they're flowering, how much flowering there is, their visual quality, and lastly, the metabolite accumulation. So I'll be using the HPLC method that I described to you earlier to extract the different pharmaceutical compounds and see if their accumulation, or see what their accumulation is. And really the main purpose of this objective is kind of to observe, to see how these plants grow in the Florida climate. And I think it'll be a good basis to see if these plants could also be an ornamental crop, because as you've seen from the pictures, they do produce at least from my perspective, quite beautiful flowers. So I think they could be a nice ornamental crop as well. So on to the ginger and turmeric research that is going on here at the University of Florida that I mentioned earlier. So ginger and turmeric are both superfoods that are edible, and they both possess some human health benefits. And they also have some ornamental value with some nice looking foliage. And another interesting point to note is that the United States is the largest importer of ginger and turmeric in the world. So part of this research is being conducted to see if we can actually produce ginger or turmeric here in Florida and kind of reduce these different prices, because it costs a lot of money to transport all of this material, likely from Asia here to the United States. So this project has 4 main research objectives. The first is to evaluate the economic yield and compare production system options of different varieties in 5 different Florida locations. So they're going to test and see if the plants grow better in the container versus directly down into the soil, or they do better covered or directly out into the field. Their second objective is to evaluate the quality of locally grown product and compare the imported product in terms of achieving market specifications, post-harvest performance, and value-added proposition for the Florida-grown plants. Their third objective is to identify size, price, and profitability of different products or of different markets for fresh, dried, and value-added products. And their last objective is to engage and communicate project outcomes with consumers, producers, and likely with Florida homeowners as well, to see if these plants are to communicate how well these plants are growing here in the Florida climate and different ways to grow them. So here's a little bit that they've shared with me about their project progress. So to start off with, they've done an invasiveness evaluation. Since these aren't Florida natives, they wanted to see, hey, if we put these out in the Florida landscape, will they become invasive? So they kind of determined they have a low invasiveness risk. They've also conducted the container production trials in the 5 Florida locations. And they've also done some photoperiod propagation and some heat stress trials to kind of see the best ways to produce these plants and grow them here in Florida. They've also conducted their phase 1 of market analysis. And so for now, they're able to start putting together how well these plants will be profitable if someone were to try to grow these for a profit. So lastly, I'd like to leave you guys with some Florida natives with some medicinal properties. I'm not very well educated on the actual medicinal properties of these plants and where they're derived, but these are all Florida natives. And so obviously they're well adapted to our climate here and would do well on different Florida landscapes. And they all possess some kind of medicinal property as well. So starting off with slash pine, and then also saw palmetto. One of the grad students here in the Environmental Horticulture Department is actually investigating saw palmetto potentially for its medicinal properties. I believe initial findings have found that it has some cancer properties or anti-cancer properties that may be attributed to the berries, but I'm not quite sure of that research just yet. And then also wax myrtle, sassafras, also are Florida natives that may have some medicinal properties. A quite nice looking plant that we see here in Gainesville and just south in our big, oh shoot, what is the area called? I'm forgetting now, but the white water lilies, they're quite gorgeous. I love the flowers these plants produce. Witch hazel, one of my favorite flowers, the purple passion flower. I personally just bought one of these my plants myself because I love how showy these flowers are. The red maple, beauty berries, and also buttonbush. So here are just a few references I used for both my research and to put this presentation together for y'all. And I'd like to acknowledge a few people. Firstly, I'd like to thank my PI, Dr. Swati Nadakadudi, for her guidance throughout my master's program and for giving me the opportunity to present to you all here today. I'd also like to thank some of our lab members, Dr. Ken Cho, a biological scientist. He's been a great help with all my experiments and has provided some great guidance. Also, undergraduate student in our lab, Megan Takev. She has been a great help with setting up a lot of my experiments and helping me process some different samples. Also, I'd like to thank Dr. Selena Gomez and some members from her lab. They've helped me tremendously with my controlled environment experiment and setting that up, setting up the lights. They've also let me borrow the blue lights that I'm using currently. And then also Dr. Paul Fisher and his group has also been a great help in giving me advice on how to just grow these plants, how to conduct the controlled environment studies as well, and different things I've needed throughout my research. They've been a fantastic help. So without all these people, I really wouldn't be where I'm at right now in my research, and they have all been a great help to me in my progress here in my master's program. So with that, I thank you all for your time, and now I think we have plenty of time to answer some questions if any of you have them.
Great, Blake. Thank you so much. That's really fascinating research you're doing, and was really interesting. And quite a few questions were coming in, so I'll go ahead and kind of read some of those off to you and let you answer them. One of the first ones was, what is the plant using the flavonoids for? I don't know if you have any idea how to answer that one.
So flavonoids and other different phenolic compounds are often used to kind of reduce these things called reactive oxygen species. So these different compounds will be produced by the plants when they're under stress. So whenever the plant kind of comes in contact with stress, it produces these harmful chemicals and it can damage the plant in a variety of different ways. So essentially these flavonoids are really potent antioxidants. So like kind of like a blueberry or something like that. And they're able to reduce these different reactive oxygen species within the plant and kind of protect it against those harmful chemicals. So it's kind of in a defense mechanism is essentially what these flavonoids are being functioned as.
Okay, fantastic. Can the Scutellaria species be found for planting in South Florida, the ones you discussed?
Yeah, I believe some of them would do well. I haven't heard too much of people actually growing them in South Florida, but I mean, personally, I think it'd be worth a shot. I know likely Scutellaria havanensis is the one from Cuba, kind of hence the name Havana. I think that one may do well in South Florida since it's found in Cuba and might be a similar climate for that species.
All right. Are the pots of the skullcaps painted white for any particular reason in the test?
Yeah, they are. That's an interesting thing to notice. So actually, Dr. Paul Fisher's group did a study where they noticed how the different colored pots are actually affecting the growth of plants. And so I know I'm putting these plants out onto a black nursery pad. And here in the Florida summer, it gets very hot. So I was a bit worried about the plants being stressed from the heat. So I figured I could paint these pots white, and hopefully that would kind of reduce the heat on these plants and kind of reduce the soil temperature. So that was kind of the purpose of painting them white. That's a very good catch.
It's very interesting. There's so many variables that, you have to take into consideration, and it's everything you do, every little thing has some effect on the research, I'm sure.
Of course, yes.
Is there a difference in flavonoid levels in lab versus wild plants?
Interesting. I would actually like to investigate that. I've gotten some leads on potentially some wild plants around Florida that I could possibly go and collect from. I'm kind of in the works and speaking to some people about that. So that is something I would like to test and see, are the plants growing in the wild actually producing more or less or different profiles of these flavonoids? So yes, it's something I'd like to investigate, and hopefully I'll be able to find that out for you.
More future work.
Yes.
Will you try different skullcap tissues to check the quality and quantity of the flavonoids?
Why?
Will you try the different tissues, the flowers,
Yeah, so that's part of why we're collecting the flowers, the leaves, the stems, and the roots. So that's kind of my initial profile, is to see, are these profiles being different in these different tissues? So kind of something I didn't mention within the presentation is, interesting enough, the bicolensis produces a different kind of flavonoids only within the roots. It does produce some different ones in the leaves and stuff, but it's particularly only found in the roots, which is a very different class of flavonoids. So that's kind of the reason why we're looking at all these different plant tissues to see where they're being localized and things like that.
Okay. Is there a specific ginger that you're testing?
Ooh, I'm not quite sure on that. I know they're using mainly an edible ginger, and I think they're also testing Thai ginger, if I'm not mistaken. That would be something I'd have to get in contact with Dr. Fisher about and get back to you on.
Okay.
Mm-hmm.
Let's see. Any benefit? Let's see. You already answered that one. Let me go back to the question and answer. Where are the 5 test growing locations in Florida?
Ooh, I'm not quite sure. I know one of them would be here in Gainesville. I believe also the Mid-Florida Research Extension Center in Apopka, I believe, I think they have a testing center down there. And there's a few other locations. I'd have to look into that one and double check on their research.
Okay. When you're cultivating these plants, are you using organic methods?
For my plant specifically, it's been very hard to use organic methods. We've had some bouts with whiteflies and aphids, and it's been quite difficult to treat those organically to get rid of these pests. So we've been using some pretty hard pesticides on them, but none of these plants are being used for any kind of human consumption. They're just for laboratory plants to be tested. So not just yet. We would like to kind of start to veer towards that and reduce the amount of pesticides and things like that we're using, but they're not very organic now.
All right. Would the quality and quantity of flavonoids be affected by stress?
Yes. So as some research has been done out there to kind of see different kinds of stress, so like water stress. If the plants experience drought, for instance, they do produce different amounts of these flavonoids. Nutrient stress, are they starved for nutrients or have a lot of nutrients? They're producing different amounts. And that's also why I'm testing the blue light, is to see, all right, well, these blue lights stress the plants and cause a difference in the flavonoids. Yes, so for sure, the stress is a big factor in the different concentrations of these.
Okay.
Mm-hmm.
How tall do the native varieties of skullcap grow?
Oh, interesting. So Scutularia, the lateriflora, the blue or mad dog skullcap, it's more of a bushier plant. It stays kind of dense and compact. This is just from my personal experience growing them out here in Gainesville. I believe Arenicola, the Florida scrub skullcap, it stays a bit smaller and lower to the ground, kind of almost a similar bushier pattern. But I've also seen with the integrifolia, the helmet skullcap, is a bit taller, and they're staying all. These plants are maybe ranging from 3 to 6 months old, staying all pretty small, compact, not too big. I think it would largely depend on what kind of conditions they're being put in and what's kind of closest to their native environment.
Have you tried growing turmeric in hydroponics or aquaponics?
I'm not sure if they've tried that yet. That would be interesting because the main, I guess, component that they're using is the rhizomes to be edible and everything. So I don't know if they've tried aquaponics or hydroponics yet. That'd be something I'd have to get with them and get back to you on.
Okay. Can you drink the tea of the Scutularia lateriflora, excuse me?
I wouldn't recommend doing that. Again, that's kind of our disclaimer. We're not here to recommend any of these plants to be consumed. That is what traditionally was used by the Native Americans, but I do not know if there's any potential allergic reactions that can happen from these, or potential drug interactions may be an important thing to consider. So I wouldn't recommend it, but it is what's been traditionally used. So.
What is the preferred soil for the 3 preferred native scutellarias?
Interesting. So the Scutellaria lateriflora, the mad dog or blue skullcap, so many names to keep in my head, is more of a marshy grown plant. So it likes like a thick kind of humus soil, likes it pretty wet. It is actually kind of commonly found here in Florida in like the ditches on the side of the road. So they like it pretty wet, very humusy. Arenicola, the Florida scrub skullcap, likes it a bit drier. It's kind of found kind of in your scrub habitat, so likely a lot of sand is in the soil. It likes probably to be dried out just a bit more. Integrifolia, I would say likely falls somewhere in between. From my experience with it, it'll take being dried out and it'll take being pretty wet. And the soil that we're using is a pretty generic nursery soil. It has, just peat and perlite and things like that in it. So it's pretty generic. So I think they'll all grow and fairly well. Some just like certain conditions a little bit better than others.
Okay.
Mm-hmm.
How heavy, or how much seed do the skullcaps produce? Are they very prolific?
Interesting. So from my experience, the Scutellaria bicolensis, this is something I've been battling with the Chinese skullcap. It has never set seed for me for some reason. I don't know if it's that it's out of its native climate since it's from China. It's never set seed, but I will say the Arenicola, the Integrifolia, and the lateriflora have all been very prolific in producing seed. So almost every flower will produce a seed pod with, I think maybe 4 seeds. And I have plants right now that are just absolutely covered in seed. So they're pretty prolific in producing seed, and likely if you put them out in the landscape, I would assume they would spread pretty rapidly.
Well, fantastic. Blake, this has been so good. You've answered so many questions and covered a lot of material. Looking at the chat, a lot of people enjoyed your program. So thank you so very much for this. I do want to remind everybody that you can tune in again on June 15th at 11 a.m. Eastern, and the topic will be attracting wildlife to your yard. Don't forget to complete the exit survey, and thank you for joining us for this webinar today. Everybody have a great day. Blake, thank you once again.
Thank you.